Good morning, Grizzlies. I'm here, Mr. Larson, and I'm here just to explain my portion of this STEM lab. I just want to remind you of a few things. First thing I want to remind you is that you are taking on the persona of someone that is going to be a botanist. Now, this botanist is in charge of designing the best scenario or the best set of circumstances, or whichever way you want to think about it, the way in which you can support life to the fullest on Mars. So, if you look down here at the bottom of the screen, it says, "You've been selected as the chief botanist in the first colonial biosphere on the planet Mars. You are responsible, yes, you are, for finding out the best conditions for supporting photosynthesis in a habit that can support life forms found on Earth." Now, we've learned about photosynthesis in class, and I just want to remind you about a couple things just so you can kind of think back what we've talked about already. But we all know that photosynthesis, and the reason I'm going over these are going to be important for my variable that I'm doing. We know that photosynthesis is going to involve chlorophyll being excited, and when it gets excited, it's going to be able to move those electrons. And so we have different types of chlorophyll pigments, chlorophyll A and chlorophyll B, and they're excited at different wavelengths of light. And so you notice here that they can be excited at the wavelengths that are in the red and the blue categories. And so if you think about this, that's going to be important for us to think about when we're going through different factors. So we have some control data for you that I'll share with you in class, and you'll get in all your other classes as well. But what I want to just remind you about is when we're talking about photosynthesis, there's different factors that are involved. Temperature, I'm not going to be testing that today, or oxygen concentration, or carbon dioxide concentration, or any of the air or water or the minerals. But what I am going to be looking at is the light quality. And so if you're thinking about the types of light, so let's say that you are trying to grow something in your room for this experiment. If we send this all home with you, you'd be really concerned about which light bulb you used. And so the what the other biology teachers gave to me was to test out to see if, because we know that red and blue are the two peaks for absorption and exciting those chlorophyll molecules, if it would be a benefit to us to use just red and blue light. And so what I've done is use the materials they've given me. I'm going to film this around so you can see what's happening. And what I'm doing right now is running an experiment. And you can kind of see right there that this experiment has the red lights around the bottom and the blue light on the top, and we're determining the photosynthesis activity of those spinach inside this chamber. You can also see my lava lamp there on the side that's providing a little bit of red and blue light as well. So it's going to be an interesting experiment where we're looking at that and trying to figure out what the data is for that. And so what I'm doing is trying to help you out to determine if the changing the light bulb, the red and the blue, that combination is going to maximize photosynthesis. So what I'm going to do is just run this data for 30 minutes, and then we'll come back with some data points for you that you can then analyze. And if you have any questions about it, then this would be a time for you to kind of think through what are the reasons for the data that we get.